Bismillahirrahmanirrahim. Starting with the name of Almighty Allah, who is the most merciful and omnipotent. I am very pleased to present before you all in International Brain and Biomedicine Conference. My name is Khizra Ashraf and I am from University of Agriculture, Faisalabad. The topic of my presentation is propolis, a useful adjuvant against neurological disorders. First of all, talking about propolis. Propolis is actually a Greek word, which means pro means defense and polis means city or community. Propolis is actually a resinous or glue-like sticky material, which is mixture of honeybees mixture that honeybees produces by mixing saliva and beeswax with the exudate gathered from tree buds, sap flows, or other botanical sources. It is used as a sealant for unwanted open spaces in the beehive. As the name indicates, pro means defense. Propolis is actually a defensive material which protects the beehives from viral or antimicrobial agents outside. Propolis has a lot of beneficial effects. Some of the therapeutic effects of propolis are listed here. It acts as anti-inflammatory, cardioprotective activities, antioxidant, anti-cancer, anti-allergic, neuroprotective activities, antiviral or anti-diabetic activities. Our concern here is to discuss about the neuroprotective effects of propolis. Propolis applications in food packaging and food industries is also increasing day by day. It is being used in spray drying, films, edible coatings, food ingredients, encapsulation, and food packaging. If we talk about the coating, it is being coated on the eggs to increase their shelf life. Some of neuroprotective effects of propolis listed here are the active components in propolis are beccarin, p-cumaric acid, artipelan C. Artipelan C is actually the major component of propolis. It acts as a neurotrophic like factors for promoting neuron growth factors, induced neuroid outgrowth. Neuroid outgrowth is actually a commonly used assay to study neuronal development and neuronal degeneration in vitro. Whenever the laboratory experiments are performed outside the cells, neurite outgrowth is being used to evaluate the stress factors and the neuronal death and the neuronal degradation, and also the stimulating and the promoting effects of some factors on neuronal cells. If we talk about the neuron growth factors, these are signal proteins essential for the growth, differentiation, and survival of sympathetic and sensory efferent neurons during development. Some propolis acts as an antioxidant. Propolis attenuate oxidative stress in neuronal cells. If we talk about oxidative stress is actually an imbalance in our body, imbalance in the oxidates. Reactive oxygen species or reactive nitrogen species, and our body's ability to scavenge them. Whenever there is a raised level of oxidants in their body, there rises condition which is termed as oxidative stress. Mitochondria, the powerhouse of our cell, is the early origin of ROS. ROS are reactive oxygen species generation during oxidative stress. Whenever there is oxidative stress, there is a damage to the mitochondria of the cell. If we treat the cell which is under oxidative stress with the propolis, there has been significant inhibition in H2O2 induced mitochondria derived ROS generation in neurons. H2O2 is also an oxidant which can cause oxidative stress and damage to the cells of neuron. If we treat those cells which are being under the stress of H2O2, the expression of mito-SOX red signal was significantly increased in neuronal cells after exposure to H2O2. Whenever the cells, whether these are neuronal cells or hepatic cells, whenever they are under stress of H2O2 or exposed to H2O2, there is an increment in mito-ROX, SOX red signals. The cells which are treated with a propolis methanol extract for 1 hour significantly reduce the immunofluorescence intensity of mito-SOX red oxidation in H2O2 exposed neuronal cells. The cells which are being treated with the propolis methanol extract for 1 hour in vitro outside the cell, the experiments were performed on mice. It was being observed that the immunofluorescence intensity of a micro-SOX red signal were significantly reduced after being treated with propolis. Thus, it exhibits that propolis has antioxidant effects. Some of the neuroprotective effects of propolis: neuroinflammation contributes to a decline in cognitive function during aging as well as in disease. It is the most common observed phenomena that there is a problem in thinking and intellectual ability in persons, and also in Alzheimer's and Parkinson's disease. As a potent activator for exacerbating neuroinflammation, some of the pro-inflammatory markers which were signatures of inflammation are interleukins 1 beta. This has been reported to suppress brain-derived neurotrophic factors dependent synaptic efficacy, resulting in cognitive impairment. As whenever there is an increment in pro-inflammatory markers like interleukins 1 beta, there is a decrease in brain-derived neurotrophic factors. Treatment with propolis significantly reversed the interleukins 1 beta induced impairment of brain-derived neurotrophic factor induced OCK expression. OCK expression, if we talk about the OCK expression are actually activity regulated cytoskeleton associated protein, is a critical immediately early gene that has been implicated in generated stable changes in synaptic efficacy. A suppression of OCK expression impairs the synaptic efficacy and memory consolidations. Treatment with propolis significantly reversed the interleukins 1 beta induced impairment of brain-derived neurotrophic factor-induced OCK expression. These observations indicate that propolis reversed the neuroinflammation-induced dysfunction of synaptic efficacy in neuronal cells. Propolis is an antidepressant. Adenocorticotropic hormone is released, which can cause the from the adrenal gland to produce the cortisol. Changes in cortisol concentration influence immunity, metabolism, growth, reproduction, depression, and anxiety, and some of the important physiological processes. Propolis has significantly inhibited the hypothalamic pituitary adrenal axis hyperactivity to mitigate increasing plasma ACTH and cortisol level. Propolis has therapeutic effects on anxiety through antagonizing the hyperfunction of HPA axis. HPA axis is actually hypothalamic pituitary adrenal axis levels, and enhancing antioxidant mechanism in brain tissues. Actually, propolis activates glucotrophic factors, which are the signals for the stimulation of HPA axis. Whenever its hyperactivity is mitigated, there is increased level of ACTH and cortisol, which can cause depression in stressed models of rats. Therefore, by altering that signals pathway, there is a decrease in depression level, and a comforting and serenity state is achieved, which kept in the right state of mind, and thus decreasing the level of depression in models under study. Neuroprotective effects of propolis. Propolis are rich in polyphenols, and some of the phenolic compounds, like terpenoids in propolis, has neuroprotective effects in many models of neurodegenerative diseases. The mechanism of this neuroprotection are complicated and associated with multiple pharmacological effects, such as antioxidant activity, anti-inflammatory activity, inhibition of excitotoxicity of neuronal cells, inhibition of cell cycle progression, and protection of mitochondrial dysfunction in ischemia. Propolis protects against the hypoxia induced microglial mediated neuroinflammation. If we talk about the microglia are resident cells in central nervous system, which are actually the immune cells for our brain and for other parts of the central nervous system. These are actually the first line defense of the central nervous system. Whenever there is a brain injury or any stress material is there, they excite the release of inflammatory markers and cause the neuroinflammation. If that stress level is not reduced, they ultimately cause the neuronal death. Anti-cancer effects of propolis. Propolis inhibit apoptosis of nerve cell by reducing the expression of genes associated with apoptosis signaling pathways. Whenever there is a level of stress and cell goes to programmed cell death, and we want to reverse that state, the propolis treatment actually reverse the signals which are responsible for ongoing apoptotic process. Propolis in vitro, as well as its protective role against methotrexate toxicity. Methotrexate is a chemical which is caused hepatotoxicity and neurotoxicity in some of the animal models. Propolis acts against that methotrexate induced toxicity. Ethanolic extract of propolis was cytotoxic to cancer cell lines and showed anti-tumor effects by reducing the volume of tumors, decreasing the count of viable cells in the mass, and prolonging the lifespan of affected subjects. Improvements in methotrexate-induced hepatorenal toxicity in animal models were also recorded. Egyptian propolis extract revealed high apoptotic effects through an increase in BAX. BAX is actually a apoptotic protein, caspase 3, cytochrome C expression levels, and by reduction in B cell lymphoma 2. And B cell lymphoma 2 is an anti-apoptotic protein in the tested cancer cell lines. Propolis also enhances the G naught and G1 cell cycle arrest induced by methotrexate, thus decreasing the level of tumor cells in cancer cell lines. That's all about the propolis. Propolis is actually a dietary material which is being used effectively in nutraceutics. If we talk about the nutraceutics, it is actually the foods which, alongside the provide additional health benefits and therapeutic effects alongside the nutritional values. That's why it is the emerging field in industry. Alongside the pharmaceutics, the protective superfoods and the magical effects of these foods on health benefits of human has increased the level of their usefulness and the level of their industrial implementation day by day. That's all. Thank you.